The overall goal of this procedure is to describe the methodology that will allow us to examine how individual neurons and local field potentials in different cortical layers of the primary visual cortex encode sensory information. The procedure begins with describing the construction of the computer-controlled microdrive system and the use of a multi-contact laminar electrode for the recording in the primary visual cortex. The next step is to perform an evoked response potential paradigm after the electrode has been advanced to the target brain region. Following this, current source density analysis is used to identify the cortical layers according to the polarity inversion accompanied by the sync source configuration. The final step of the procedure is to perform receptive field mapping and analyze the differences in neural activity in response to visual stimulation. Ultimately, results can be obtained that show layer-specific changes in encoding sensory information.
Hi, my name is Sarah Eagleman, and I'm a graduate student at the University of Texas Medical School in Houston. The main advantage of this technique over existing methods, like multi-electrode arrays, is that the U-probe can record neural activity across many millimeters of cortex simultaneously in a single penetration.
Hi, my name is Bryan Hansen, and I'm a graduate student working at the University of Texas Medical School in Houston. This method can answer key questions in the neuroscience field, exploring whether and how information is processed in a laminar-specific manner.
To construct the Nein electrode drive assembly, first assemble the necessary tools and pieces, including the guide tubes, guide wire, complete Dremel set, Nein tools and parts,